Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience-based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
What if I told you there's like a hidden range extender built right into our DNA?
A range extender.
Yeah, a secret switch, kind of, that lets these genetic elements called enhancers turn genes on from, well, frankly, staggering distances. We're talking hundreds of thousands, maybe even 1 million base pairs away.
Okay, that's pretty far.
Right. And here's the really weird part. This range extender seems to work totally independently of what the enhancer actually does, or even how the DNA is folded up in 3D space.
Huh. That definitely goes against some of the standard thinking.
Welcome to the deep dive. Today, we're digging into a really fascinating 2025 Nature paper. It's titled Range Extender Mediates Long-Distance Enhancer Activity.
Sounds intriguing.
Our mission, as always, is to pull out the key insights from this research, cut through the dense scientific language, and give you a shortcut to understanding a really fundamental piece of how our genes are controlled.
It's a great topic because, for years we've understood that enhancers often act nearby their target genes, maybe tens of thousands of base pairs away. That's sort of the standard model.
Right, the local action.
Exactly. But then you have these outliers, these exceptions like the famous ZRS enhancer. It's critical for limb development.
Ah, yes. The ZRS controls the sh gene, right?
Yeah.
Sonic Hedgehog.
That's the one. And it controls shift from, get this, about 848,000 base pairs away in mice. A huge distance.
Wow.
So how these extreme long-distance interactions actually work, that's been a major puzzle in gene regulation for a long time.
Totally. And for a while, the main explanation people focused on was the 3D structure of the genome.
Right. Things like CTCF proteins, cohesin, these topologically associating domains, TADs. The idea that the DNA physically loops around to bring distant elements close together.
Makes sense. But as we've seen in more recent work, even when you disrupt those 3D structures quite severely, gene expression often holds up surprisingly well. It's more robust than you might expect.
Which really raises the question, okay, if it's not just the 3D folding, what else is going on?
Exactly. What other factors, maybe encoded right in the DNA sequence itself, are setting up and maintaining these crucial long-range communications, especially during development? And that's what this paper tackles head-on, using some really elegant experiments in mice.
Okay, so where do they start?
Well, they kicked off with a really basic but fundamental question. Can any old, good limb enhancer just substitute for the ZRS? Can it do the job at that extreme 848 kilobase distance?
Or is there something special, something inherent about the ZRS that lets it work so far away?
Precisely. So they did a replacement experiment. They took out the ZRS in mice and swapped in 4 other well-known strong limb enhancers.
And these weren't just random enhancers.
No, these were proven players. HS72, which normally works from 411 kilobases away. MM1564 from 177 kilobats. HS1516 from 147 kilobats. And MM1492, the closest, working at 73 kilobats. So a range of native distances.
Okay, so they put these known enhancers in the ZRS spot, far away from whatever.
Well, here's the kicker. Despite being perfectly good enhancers in their normal homes, all 4 of them completely failed to turn on from that long distance.
Failed how? Like weak expression.
No, like no expression. The mice ended up with severely shortened, truncated limbs, basically indistinguishable from mice that completely lacked function in their limbs.
Wow, okay, so that's a clear result.
It really is. It powerfully demonstrates that just being a strong, tissue-specific enhancer isn't enough. There's something else required to bridge that huge genomic gap.
But, being scientists, they had to be sure it wasn't something trivial, right? Like maybe the enhancers got shut down in the new location. Or maybe they just didn't play well with the promoter specifically.
Exactly. Classic potential pitfalls in these kinds of experiments. So first, they checked accessibility. Were the enhancers even open for business at the new spot?
Using something like ATAC-seq, I assume?
You got it. Allele-specific ATAC-seq. And the answer was clear. Yes, the transplanted enhancers were open, accessible. They weren't being silenced epigenetically.
Okay, so rule out silencing. What about promoter compatibility? Maybe these enhancers just can't talk to the promoter.
Good question. They tested that directly, too. They took one of the enhancers, HS72, and put it really close to the promoter, just 2 kilobases away in a different mouse model.
And what happened then?
Then it worked perfectly. HS72 activated strongly at close range, so strongly, in fact, that the mice developed extra digits polydactyly.
Ah, so HS72 can activate no problem there. It just couldn't do it from 848 kilobases away.
Precisely. It proved the enhancer and promoter were compatible. The issue wasn't the enhancer's intrinsic ability or compatibility, it was purely the massive distance.
Okay, so the mystery deepens. The enhancers are open, they're compatible, but they can't shout loud enough to be heard from that far away.
Right, which led them to hypothesize. Maybe there are other sequences, specific bits of DNA that are associated with these naturally long-range enhancers. Something extra.
A missing piece needed for long-distance communication.
Exactly. And so they started looking. They focused on the region around HS72 because that was the one that worked from the furthest native distance among the failures, remember, 411 kilobead.
And did they find something?
They did. They identified this highly conserved stretch of DNA nearby, and they gave it a great name, REX, for a range extender.
REX. Okay, I like it. Does REX itself act as an enhancer?
That's the crucial point.
Yeah.
No, by itself, REX has no detectable enhancer activity. It doesn't turn genes on.
Okay, so what happens when you put REX with an enhancer?
Now we get to the breakthrough. They took HS72, the enhancer that failed at long range, and they physically linked the REX sequence to it. They made an HS72 plus REX combo element.
And transplanted that to the distant locus.
Exactly, and this time, it worked. The HS72 plus REX combo successfully activated expression from 848 kilobases away, and the mice developed completely normal limbs.
Wow. So REX rescued the long-range activity of HS72?
Completely rescued it, but wait, it gets even better. They then took the shortest range enhancer they'd tested, MM1492, the one that normally only works from 73 kilobases away.
Okay.
They added REX to that one, creating MM1492 plus REX, and they put that combo at the distant locus.
And did it work? From 73 kilobar native range all the way to 848 kilobar.
It did. This MM1492 plus REX element also induced expression and led to fully developed limbs.
That's incredible. So REX could boost the range by more than tenfold in that case? From 73 kilobars to nearly 850 kilobars.
Exactly. It's like REX is a modular turbocharger you can just bolt onto an enhancer to give it long-distance capability.
Which beautifully demonstrates that REX seems to be both necessary because HS72 alone failed, and sufficient because adding it rescued HS72 and even boosted MM1492 for this long-range function.
Right. It acts like a separate module.
So what's inside REX? What's the actual sequence doing the work?
Good question. They zoomed in on the REX sequence itself, looking for known patterns, specifically binding sites for transcription factors.
Makes sense. What do they find?
They found these highly conserved motifs, short DNA sequences, specifically C-tata homeodomain motifs.
Ah, homeodomain motifs. Those are known binding sites for a class of developmental regulators.
Precisely. And these particular motifs matched the binding preferences for transcription factors called LHX2 and LHX9, which, crucially, are known to be important in limb development.
Okay, so there's a plausible molecular connection there. LHX factors binding to these motifs within REX.
Plausible, but they needed to prove it. So they did the key experiment. They mutated those specific C-tata motifs within the REX sequence.
Mess up the code and see what happens.
Exactly. And when they mutated those LHX motifs in the HS72 plus REX construct.
Let me guess. The long-range activity was lost.
Gone. Abolished. The mice had truncated limbs again. But, importantly, if they mutated a different predicted motif in RX, one for a factor called LAF1, it had no effect.
Okay, so it's specifically those C-tata motifs that are essential for REX's function.
Seems so. And it gets broader. They looked genome-wide at other known limb enhancers.
To see if this was a general pattern.
Yep. And they found that these exact same C-tata motifs are significantly more common, enriched, in long-range limb enhancers, the ones acting over hundreds or thousands of kilobases compared to short-range ones.
Interesting.
And even more, having multiple copies of these motifs seem to correlate with even longer operating distances.
So it suggests this isn't just about RX, but maybe a general sequence signature, a code for long-range enhancers in the limb.
Exactly. Which brings us back to the original long-range champion, the ZRS itself.
Right. If these motifs are the key, does the ZRS have them too?
Excellent question. And yes, it turns out the ZRS naturally contains 4 of these citata motifs embedded within it.
Okay, so the natural enhancer uses the same code.
Seems like it.
Yeah.
So the researchers did the definitive experiment. They used CRISPR gene editing to mutate just those 4 motifs within the natural ZRS in mice, leaving the rest of the ZRS intact.
Wow, precise editing. What was the result?
The result was striking. Mice with just those 4 motifs mutated in their ZRS developed severely truncated limbs, identical, really, to mice completely missing the ZRS, or lacking SH function.
So those motifs are absolutely critical for the ZRS's natural long-range function.
Absolutely critical. But here's maybe the most illuminating finding of the whole study.
Okay.
Even with those 4 essential long-range motifs mutated, the ZRS still worked perfectly fine at short range when tested in reporter assays.
Ah, so you can separate the two functions.
Exactly. It clearly demonstrates that the ZRS's ability to specify limb expression, its tissue specificity, is a separate, distinct function from its ability to project that activity over a huge distance. You can break the long-range communication without breaking the core enhancer message itself.
That's a really fundamental insight. The what and the how far are encoded differently.
Totally uncoupled. And the final piece, they took that mutated ZRS, the one that couldn't work long range anymore, and they added the REX element to it.
Could REX rescue the mutated ZRS?
Almost perfectly. Adding RX back restored the long-range activity and led to almost complete rescue of limb development.
Which proves that the citata motifs in RX are functionally doing the same job as the ones naturally found in the ZRS. They're interchangeable parts for long-distance signaling.
Exactly. It provides this really compelling model where these specific motifs, likely bound by LHX factors, are the key mediators of extreme long-distance enhancer communication, at least in the developing limb.
So stepping back then, what's the big takeaway here? This really shifts the focus, doesn't it?
I think so. It reveals this fundamental mechanism encoded directly in the DNA sequence that explains how enhancers can bridge these enormous distances. It's a big step beyond just thinking about 3D folding.
Absolutely. The real innovation is identifying this portable sequence code, the RX element, defined by these citata motifs that seems to act like a dedicated switch or a booster for an enhancer's range.
And it decouples things so nicely. The enhancer's job, like activate SH in the limb bud, is separate from its reach, do it from 850 kilometers away.
Right. It gives us this new design principle almost. You have your core enhancer for specificity, and then you potentially add these motifs to dictate the range.
Add motifs, extend range. It sounds so simple, but it's powerful.
It really is, and it opens up so many questions for future research, of course. We still need to figure out the exact molecular mechanism, what proteins are actually bridging the gap. How do these motifs and the factors that bind them interact with the known machinery like cohesin and loop extrusion? Is it facilitating looping, or is there another mechanism?
And how common is this? Is REX specific to limbs, or are there similar range-extending elements with different motifs in other tissues, other developmental contexts, or even other species?
Exactly. Is this a universal principle of genome organization, or more context-specific? Lots to explore there.
And the implications for human health could be pretty significant, couldn't they? When we think about genetic diseases caused by changes in non-coding DNA.
Definitely. You can imagine scenarios where a mutation doesn't break the enhancer itself. It might still work fine locally, but it specifically disrupts these range-extending motifs.
So it loses its long-range capability, leading to disease, even though the core enhancer sequence looks okay.
Precisely. Understanding this distinction, this modularity, could really change how we interpret non-coding variants found in patients with developmental disorders or even cancer susceptibility. It might lead to better diagnostics and potentially new therapeutic ideas.
What a fascinating journey this paper takes us on. From seeing enhancers fail unexpectedly at long distances to finding this tiny REX element, pinpointing specific motifs within it, and then showing those same motifs are key to how the natural ZRS works over nearly 1 million base pairs.
It's a really elegant piece of science. It provides a crucial new layer to understanding the complex regulatory language written in our genome. We're moving beyond just knowing what elements do to understanding how far their influence can stretch.
It really changes the picture. So here's something to think about, a provocative thought to leave you with. If we can now understand, and maybe even engineer, an enhancer's range by tweaking these motifs, What are the possibilities?
Hmm.
Could we potentially design synthetic enhancers for gene therapy that are engineered to be super potent, able to reach and activate target genes from much further away than natural enhancers typically do? Could that lead to more effective therapies? And beyond range, what other hidden codes are embedded in our DNA, controlling fundamental processes in ways we haven't even conceived of yet?
It certainly opens the door to thinking about genome regulation in a much more nuanced, sequence-driven way. Lots of exciting possibilities ahead.